Good afternoon. In previous class, we have discussed about the what are human physiology and anatomy, and followed by the sodium potassium pump and action potential and resting potential. In this class, we are going to discuss about the sodium potassium pump. The sodium potassium pump is a protein that is found in the plasma membrane of the cells. It is responsible for maintaining the concentration gradient of sodium and potassium ions across the membrane. The pump uses ATP energy to pump 3 sodium ions out of the cells for every 2 potassium ions pumped into the cells. Electrodes are used to measure the activity of the cells, including the sodium potassium pump. There are two types of electrodes, microelectrodes and macroelectrodes. Microelectrodes are very small and are used to measure the activity of individual cells. Macroelectrodes are larger and are used to measure the activity of the group of the cells. These electrodes can be used to measure the electrical activity of the cells, such as resting potential and the excitation potential, which are discussed in the previous class. And what is electrocardiogram? An electrocardiogram is a test that measures the electrical activity of the heart. It is a non-invasive test that involves placing electrode on the skin of the chest, arms and legs. The electrodes are connected to an ECG machine which records the electrical signal produced by the heart. The ECG machine produces a graph which shows the electrical activity of the heart over time. The graph shows different stages of the cardiac cycle, including the P wave, QRS complex wave and T wave. These waves represent the depolarization and repolarization of the atria and ventricles of the heart. We can see a block diagram of the ECG, which includes the protection circuit and lead selector. Through the lead selector, we can get two circuits like a fault detector and calibration circuit, through which we can connect the instrumentation amplifier, through which the amplifier we can connect to the filtering to filter the unwanted signals from the heart, and also to the final amplifier to induce the low frequency into the high frequency, through which we can connect it to the oscilloscope, in which the oscilloscope is consist of driver amplifier, recorder and a pen motor. The pen motor is the main component which draws the graph of the electrical signal coming from the heart, which in turn we can read it as the PQRST wave. An ECG, the four waves in an ECG are P wave, QRS complex, T wave and U wave. And P wave, this is the first wave in the ECG and represents the depolarization of the atria. It is a type typically small and rounded, which we can represent in the figure. And the QRS complex this complex consists of three waves, Q wave, R wave and S wave, and represents the depolarization of the ventricles. The QRS complex is usually the largest wave of the ECG. This wave and T wave, this wave represents the repolarization of the ventricles. It typically is slightly rounded wave that follows the QRS complex. And U wave, this wave is less commonly seen in the standard ECG and represents the repolarization of the Purkinje fiber in the ventricles. The U wave is typically a small rounded wave that follows that of the T wave. Next, we can see the electroencephalogram. An electroencephalogram is a medical test that measures the electrical activity of the brain. It involves placing small flat metal electrode on the scalp to detect the electrical impulses produced by the brain's neurons. These impulses are then recorded and analyzed by a computer to create a visual representation of the brain's electrical activity, which can be used to diagnose various brain disorders. And an EEG is a non-invasive and painless test that typically takes around 30 to 60 minutes to complete. During the test, the patient may be asked to lie down and relax with their eyes closed or open to perform certain tasks or activities to simulate the brain activity. The recorded EEG can then be analyzed by a trained healthcare professional to detect any abnormal patterns or activity that may indicate a brain disorder, such as epilepsy, sleep deprivation, or brain injuries. EEG is widely used as a diagnostic tool in neurology and psychiatry and can provide valuable insights into brain function activity. And we can see the block diagram of it, through which we can get a clear view, in which the scalp electrode, which we have placed in the head of the patient, through which we can connected to the instrumentation amplifier, through the active high pass filter, through the high pass filter, low pass filter and notch filter, through which we can connect it to the post amplifier. Through the post amplifier we can connect it to the DAC card and to the computer for the display, through which we can able to describe the particular output. The output graph of an EEG typically consists of series wavy lines known as brain waves. These brain waves are divided into several frequency bands which are measured in hertz or cycles per second. The frequency waves are delta waves, theta waves, alpha waves, beta waves and gamma waves. The waves varies from 0.5 to 4 hertz. These waves are the slowest and represent deep sleep or unconsciousness. Theta waves, which includes the range from 4 Hz to 8 Hz. These waves are associated with the drowsiness, daydreaming and light sleep. Alpha waves, which in the range of 8 to 12 Hz. These waves present when the eyes are closed and the person is relaxed or meditating. Beta waves is in the range of 12 to 30 Hz and present when the person is alert and focused, and can be further divided into low beta waves, which ranges from 12 to 15, and high beta waves ranges from 15 to 30. And gamma waves are within the range of above 30 Hz. These waves are associated with the high levels of concentration, problem solving and cognitive processing. In next class we can see about the EMG, that is electromyogram. Now, thank you.